In last lecture, we have started Monida. Okay, students. So today I will give you the diagram of a typical bacteria. Suppose this is the bacteria. Now, outermost layer is Glycocalyx. Second layer, cell wall. Third layer, plasma membrane or cell membrane. Okay, plasma membrane or cell membrane. Now, inward invagination of plasma membrane is called mesozome. Suppose this plasma membrane has inwardly invaginated like this. This structure is called mesozome. Now, with this mesozome, actually, nucleoid material attaches. Now, this is the nucleoid part. This is circular DNA, which is called plasmid. I will discuss each and every structure in detail, student. Here, just I am focusing on the diagram. Okay. So, extra chromosomal material, that is plasmid. Some storage materials are also present. They are called inclusion bodies. Okay. With green I am drawing cytoplasm. And with yellow I am showing ribosomes. Ribosome is called protein factory, students. Yeah. Some locomotory organ are also present, some appendages. Suppose which are longer in structure. Okay. They are called pili or pili. And which are shorter in structure, they are called fimbri, like this. They are fimbri. Now, fimbri is more in number, and pili are less in number. Now, diagram is clear to you, student? Diagram of a typical bacteria. Diagram is clear to you, all of you? Students?
Yes, ma'am. Yes, ma'am.
This diagram is given in NCERT also. Okay. Now, one by one, now I will discuss the features. The first one which I will discuss the features, that is for Glycocalyx. Glycocalyx. Now, these Glycocalyx are of two types of student. Okay. One is called slime layer. The Glycocalyx which are thin, which are loose, and they are made up of, MO means made of polysaccharide. Polysaccharide. They are called slime layer. Okay. And second one is capsule. Now, this capsule part is present in some bacteria. Not all bacteria presents capsule parts. Now, the glycocalyx, which are thick, which are rigid, and they are made up of polysaccharide as well as polypeptide. They are called capsules. Polypeptide means they are made up of amino acids. AA means amino acids. Now, this glycocalyx is formed by plasma membrane. Now, what is its function? Number one is protection. Okay. Now, capsule. This capsule protects from WBC. Protects from WBC and they help in colony formation. These are three important functions. Colony formation. So this is what about glycocalyx. Clear with glycocalyx, students? Clear with glycocalyx?
Yes, ma'am.
Second structure is cell wall. Okay. Now, here cell wall is made up of a structure called peptidoglycan. In short, I use PDG everywhere. The next lecture, I will use PDG. Okay. Now, only a cell wall-less bacteria, cell wall is absent in mycoplasma. Mycoplasma. Now, this peptidoglycan, they are made up of peptide. Peptide means they are made up of peptide. Peptide means amino acid. Glycan means sugar. Peptido means they are made up of amino acid. Glycan means sugar. Okay. Glycan means sugar. So what is the sugar component, student? Sugar component is NAG and NAM. What is the full form of NAG and NAM? Full form of NAG is N-acetyl glucosamine. Full form of NAM, N-acetyl uramic acid. Okay, this is about cell wall. Third is plasma membrane or cell membrane. Cell wall is clear to you? Student, cell wall. Plasma membrane or cell membrane. Now, this plasma membrane is similar to the plasma membrane of eukaryotic cell. Similar in the sense like they are lipoprotein in nature. Okay, they are lipoprotein in nature. Plasma membrane or cell membrane. They are similar to plasma membrane of eukaryotes. They are lipoprotein in nature. Lipo means lipid part, protein. That means lipid is protein is attached with lipid. Okay. Next one, mesosome. As I already told you, inward invagination of plasma membrane is called mesosome. Other name of mesosome is chondroid. Other name of mesosome is chondroid. And it was discovered by F. James. It was discovered by F. James. Now, two types of mesosomes are there. Number one is central mesosome. Okay. Now, central mesosome means they are associated with nucleoid. They are associated with nucleoid. That means in diagram which I have shown you, that is central mesosome. What is their function? They play an important role in DNA replication. DNA replication and cell division. And cell division. The second one is lateral or peripheral. Now, what is this lateral and peripheral means? They are associated with respiratory enzymes. They don't possess mitochondria, so they are playing the role of mitochondria associated with respiratory enzymes. Now, which enzyme is present in mesosome? That is very important from your exam point of view, students. In NEET, many times they have asked this question. Which enzyme is present here? Succinate dehydrogenase. Succinate dehydrogenase and cytochrome oxidase. These are the two enzymes which are present in mesosomes. Okay. They have important function in cell wall formation also. Cell wall formation, one function. And you can say that they are analogous to mitochondria. They are analogous to mitochondria. So this is what about mesosomes. Student. Any doubt in mesosome? Students, any doubt in mesosome? Any one of you? Let us see some more features. Some more features. Membrane bound organelles are absent. Okay. Ribosome present, that is for 70 years. Cyclosis absent. What is cyclosis? Cyclosis means movement of cytoplasm or cytoplasmic streaming. Okay. So movement of cytoplasm is absent. What about the genetic material? What about genetic material? Double stranded, circular, naked DNA. Now, nuclear membrane, nucleolus, histone protein, true chromosome, all these things are absent. Okay. So what is present here? Their RNA is present, non-histone protein is present. That is why the genetic material of bacteria is also called incipient nucleus, or it is called nucleoid. Okay. In some cases, it is also called genophore or prochromosome. This is about their genetic material. In diagram, I have taught one extra nuclear material is also present. What is that extra nuclear material, students? Extra nuclear material, that is plasmid. Okay, that extra nuclear material is plasmid. They are also small, double-stranded, circular DNA. They are extra chromosomal DNA. Extra chromosomal DNA. They are present in some bacteria and they are non-essential element. Why non-essential? If it could be essential, then it should be present in all bacteria. That is why it is a non-essential element, because it is extra nuclear material. One nuclear is present in all bacteria. Okay. Now 4 types of plasmids are there. 4 types of plasmid. Number 1 is R plasmid or R factor. R means what? This R means resistance or resistant. So they provide resistant against and UV rays. Okay, this is about R plasmid. First type of plasmid, that is R plasmid. Till this any doubt, any one of you students? Till this any doubt, any one of you students? Now second one is TI plasmid. Very important, student, from question point of view. You need a lot of question I have asked with this TI plasmid. First of all, you should know the name. What is TI? TI means tumor inducing plasmid. Second question, from which bacteria you are isolating this plasmid? The name of bacteria is very important. That is Agrobacterium tumefaciens. Agrobacterium tumefaciens. Now in plant, they cause a disease called crown gall disease. What is this crown gall disease? In plant, a large tumor formation takes place. The disease is called crown gall disease, and that is why the name of the plasmid is TI plasmid. That is tumor inducing plasmid. Okay. Now third category of plasmid is sex plasmid or F plasmid or F factor. What is this F? This F stands for fertility. Now again, this F plasmid are of two types. One is integrated. What is the meaning of integrated? Integrated means this plasmid is associated with nucleoid. Okay, plasmid associated with nucleoid. Now, when plasmid is associated with nucleoid, this type of plasmid has a special name. It is called epizome. It is called epizome. And second one is free plasmid. Free plasmid means they are not associated with nucleoid. They are not associated with nucleoid. Okay. And fourth category is degradative plasmid. Fourth category is called whole plasmid. Whole plasmid. Again, this whole means polysins. What are these polysins, student? It is nothing but they produce toxin so that they can kill some other bacteria. Two types of toxins they are producing. One is called polysin and other is called bacteriocins. Okay, that means they kill another sensitive bacteria. They kill sensitive bacteria. And one special type of plasmid, student. Okay, they are called degradative plasmid. What are these degradative plasmid? It was discovered by Dr. Anand Mohan Chakravarti. From one bacteria, it is isolated. The name of the bacteria is Pseudomonas lutida. This name is very important, student. From this name, you will get the question in your NEET. Okay, Pseudomonas lutida. Fine. What is their function? You know, they degrade hydrocarbon. Okay, they degrade hydrocarbon, petroleum. Now, what are present in petroleum, student? Camphor is present. Xylene is present. Octane, naphthalene. All these things are present in petroleum and they degrade this hydrocarbon petroleum. They means I'm talking about Pseudomonas lutida. Okay. Now, why this plasmid is of much importance, student? Okay. You know, in ship oils are transported, student, in large quantity. Fine. So that oil spilling takes place during the transfer. Suppose this is the ocean, or river, whatever. Okay. And here it is the ship. And with this ship, student, this oil leakage takes place. And due to this oil leakage, suppose due to this oil leakage, this oil will create a layer. Suppose this is the layer of oil above the water. So what will happen, student? This aquatic ecosystem will be hampered because layer of oil is present means what? It will not allow exchange of gases. Okay, oxygen will not enter into the water, and aquatic plants and aquatic animals will not get oxygen due to this layer of oil. So we can say now that this oil is actually creating a pollution. Which type of pollution? Oil pollution. Okay. So with the help of this plasmid, we can control oil pollution. We can control oil pollution. So we are utilizing microbes for the control of oil pollution. This type of technique is called bioremediation. This type of technique is called bioremediation. Bioremediation means, student, control of microbes. Sorry, utilization of microbes. Utilization of microbes for control of oil pollution. It's called this method, this technique is called bioremediation. So this plasmid, degradative plasmid, play a vital role in bioremediation. Clear with this, all of you? All types of plasmid is clear to you? All types of plasmid is clear to you, students? Students, any doubt in types of plasmid? Till now, any doubt? Students, any doubt? Till now, any doubts? Any one of you? Next one is outgrowth of plasma membrane. That is, I'm talking about pili and fimbriae. Okay. Outgrowth of plasma membrane. Fimbriae and pili. Now, fimbriae is thinner, whereas pili are thicker. They are smaller in number, smaller in size. They are longer in size. They are present around 300 to 400 in one cell and 1 to 5 each cell. This is about pili and fimbriae. Now, next one is inclusion bodies. So, what are inclusion bodies, student? Okay, inclusion bodies means nothing but they are storage material actually. Okay, inclusion bodies means they are storage material. Okay, they are not bounded by any membrane and they are present in cytoplasm. So they are present in cytoplasm. They store reserve material. They are not bounded by any membrane. Let us see some example of inclusion body, student. Glycogen granules. What is the function? Glycogen with name you can understand that they stores carbohydrate. They stores carbohydrate. The second one, volutine granules. Volutine means they store phosphate. So these are the inclusion bodies. Okay. Now, next we will see shapes of bacteria, student. We know that rod shaped bacteria are there, copper shaped bacteria are there. So what are the shapes? Let us discuss now. We'll talk about shapes of bacteria. Okay, so basically three categories they are divided. One is coccus, second one is bacillus, and third one spherical, fourth one spiral. Okay. So in coccus, you'll see, suppose if one single coccus bacteria, this type of bacteria is called monococcus. Okay. Two coccus bacteria, then it is called diplococcus. If it is in group, for group, we are using the word strepto. Okay. Streptococcus. And if it is in bunch form, like this, it is in bunch form, then it is called staphylococcus. Staphylococcus. Bacillus means rod-shaped bacteria. Okay. Bacillus means what? Rod-shaped bacteria. Suppose if it is single rod, then it is called monobacillus. Okay. If it is two bacillus are there, then it is called diplobacillus. In straight chain form, then it is called streptobacillus. Okay. And if in the form of stack like this, they are called palisade. They are called palisade. Spiral form, student, like this in the spiral form. Okay. And one more shape is there, one comma shape. Vibrio. It is called Vibrio. They are actually comma shape. Vibrio from name you can understand now. Comma shaped bacteria means Vibrio cholera. Vibrio cholera. Positive agent of cholera. Okay. So these are about the shapes of bacteria. Okay. Next, we will have a discussion for cell wall structure. Cell wall of bacteria in detail. Basic, I have told you about cell wall. In detail, we will see how the linkages are present in gram-positive bacteria or in gram-negative bacteria. Okay. Cell wall composed of peptide part as well as the glycan part. That we already know. NAG and NAM is the glycan part. But what are the amino acids which are present in the cell wall? So this is the cell wall. So this is peptide part and glycan part. Glycan I have already told you, that is NAG and NAM, N-acetyl glucosamine and N-acetyl muramic acid. Now in peptide bond, which are the amino acids? The name of the amino acids are very, very important for your NEET student. Okay. What are the names? One is L-alanine. Second one is D-alanine. Third one, D-glutamic acid. Fourth one, L-lysine. And fifth amino acid is DAP. What is the full form of DAP, students? Di-amino 5-malic acid. Okay. Di-amino 5-malic acid. Di-amino 5-malic acid. So these are the 5 amino acids. Now next we will see the cross linkage student. How cross linkage is present in gram positive bacteria as well as gram negative bacteria. see in gram-positive bacteria. Wait a minute, students. Gram-positive bacteria. Okay. First, let me draw the alternative arrangements of glycan sugars. This is 1, 2, 3. Okay. Alternate arrangements. What are these, students? These are NAG, NAM, NAG. This is NAG, so it will be NAM. Alternative arrangements. Huh? Sugars are alternatively arranged. NAG, NAM, then again NAG, NAM, NAG. These are the alternative arrangements of sugar. So again, tetrapeptide chain is there, students. So what is that tetrapeptide chain? Let us see. Suppose 1, 2, 3, 4. Tetrapeptide chain. Okay. Now, first one, then alternative. Second one is blank. Second one, 1, 2, 3, 4. This 4 is what, student? This 4 is tetrapeptide chain. What amino acids are present here, student? L-alanine, D-alanine, D-glutamic acid, and D-lysine. This is tetrapeptide chain. Now, this tetrapeptide chain is linked with its tetrapeptide chain. This one. Same here also. This one. Tetrapeptide chain. Same composition, student. Alanine, glutamic acid, and lysine. Okay, but they are not directly linked, student. These tetrapeptide chains are linked with a pentapeptide chain. See the structure? They are linked with 1, 2, 3, 4, 5, like this. They are linked with a pentapeptide chain. So this is pentapeptide chain. Same here also. 1, 2, 3, 4, and 5. Okay, they are not directly linked. So we can say here backbone part is sugar. Sugars are the backbone. And they are alternatively arranged. This is actually what I have shown here, the cross linkings of peptidoglycan. Here with this cross linking of gram positive bacteria. Now it differs in gram negative bacteria. How it is different? I am showing it to you. This one is clear. Cross linking of gram positive bacteria. Students, is it clear?
Ma'am.
Okay. Now see how it differs in gram negative bacteria. Suppose this is gram negative bacteria. Okay. So, same sugar arrangement, NAG, LAM, NAG, NAM, same sugar arrangement. Difference in the peptide arrangement is different. Like suppose 1, 2, 3, 4, like this now. This is the tetrapeptide arrangement. It's 1, 2, 3, 4. They are directly linked. Same here also, students. 1, 2, 3, 4, and 1, 2, 3, 4. Okay, means they are directly linked. Direct linkage is there. In case of gram-negative bacteria, direct linkage is present. This is NAG, NAM, same alternative arrangement of sugar, NAM, NAG, and here direct linkage with the tetrapeptide chain. So these are the tetrapeptide chain. Some more differences we will see regarding gram-positive and gram-negative bacteria. Okay. Let us see some more differences. Gram-positive and gram-negative bacteria. First one, from the diagram, we can conclude that in gram-positive, indirect linkage. Okay. In gram-negative, direct linkage. Okay. Here, number of linkage is more. Number of linkage is more. Here, number of linkage is less. Okay. They are thicker. Thicker in the sense like in gram-positive bacteria, peptidoglycan is thick, around 150 to 200 angstrom. And they are thinner, like 70 to 120 angstrom. And they are made up of single layer. Their cell wall is made up of single layer. They are made up of 1 to 2 layers. What are those 2 layers, student? The layer of peptidoglycan is common to both of it. Now, extra layer, that is the LPS layer. What is the full form of LPS? Lipopolysaccharide layer. Lipopolysaccharide layer. This extra layer is present in gram-negative bacteria. Okay, so we can say now that their cell wall is homogeneous. Homogeneous why? Because made up of single layer. Their cell wall is homogeneous in nature. And their cell wall is heterogeneous in nature. Heterogeneous in nature. See the diagram of gram-positive and gram-negative bacteria. Now, cell wall is homogeneous in case of gram-positive bacteria. Suppose this is the plasma membrane, and in this plasma membrane, these fatty acids are arranged here. These fatty acids are arranged here. Fine. These are the fatty acids. Now, in this fatty acid, one extra acid is present in their membrane. The name of that acid is teichoic acid. Teichoic acid is present in gram-positive bacteria. This teichoic acid contains carbohydrate, alcohol, phosphate. Okay. And these basically teichoic acid is acting as a surface antigen. What about gram-negative bacteria? In gram-negative bacteria, one additional layer is present. Just now I told you that is the lipopolysaccharide layer. Suppose this is the layer of lipopolysaccharide, lipid bilayer. This is one layer of fatty acid. This is another layer of fatty acid. Together they are called lipid bilayer. This is the cell wall part. This is called LPS. This is lipopolysaccharide. Or you can say it is the outer membrane. Okay. Then after that, there is a layer of peptidoglycan. Okay. and then again the layer of plasma membrane, or you can say it is the inner membrane. Again plasma membrane, same arrangement of fatty acid, same arrangement of fatty acid, like this, same arrangement of fatty acid. So in this lipopolysaccharide layer, lipid bilayer is present. Lipid bilayer is present. Protein is present, and lipopolysaccharide is present. Lipopolysaccharide is present. Protein, I have not shown protein in the diagram. Let me show you. This type of channels, proteins are present. Okay. This type of proteins are present. Yeah. So this is the, and one more difference is present here. Teichoic acid is present, but here teichoic acid is absent. And here one additional layer is present, LPS. This LPS is acting as a surface antigen. Okay. Here teichoic acid is acting as a surface antigen. Surface antigen. Here this LPS is acting as a surface antigen. Let us see some more differences. Gram positive and gram negative bacteria. Now, in gram negative bacteria, some channels are present. Just I have shown you now some channels. Okay, these channel proteins have large size diameter. Okay, they have large size diameter, and these are called porins. These proteins are called porins. Okay, they are hydrophilic in nature, students. Hydro means water, philic means love, means they love water. They are low molecular weight proteins. They are low molecular weight proteins. But this type of channel protein is absent, or porin protein is absent in gram positive bacteria. And last differences, student, see, they don't have the layer of lipopolysaccharide. They have the only layer of peptidoglycan. So if you will treat with enzyme lysozyme, okay, this lysozyme enzyme will digest the layer of peptidoglycan in case of gram positive bacteria. Okay. Now the resultant structure is called protoclast. Peptidoglycan layer is digested. The resultant structure is called protoclast. But due to LPS layer in gram negative bacteria, complete digestion of peptidoglycan could not take place. Why? Because LPS layer is there. So partially digested cell. You will get here. You are treating here with lysozyme. Okay, treating here with lysozyme. You are getting partially digested cell. Now this partially digested cell is called spheroplast. The spheroplast you will obtain in case of gram negative bacteria, and protoplast you will get in case of gram positive bacteria. So this is what about the differences between gram positive and gram negative bacteria, as well as we have seen the cell wall composition also. Still here, any doubt, any one of you? Students? Still here, any doubt, any one of you? Any doubt, please ask me.
No, ma'am.
So what we have seen in today's class, student, we have seen the structure of a typical bacteria. Okay. Then we have gone through the structures in detail, like glycocalyx, cell wall, plasma membrane, mesozone, as well as we have seen their functions. Okay. We have seen the 4 types of plasmids, student, the shapes of bacteria. Then what we have seen, the cell wall composition of gram positive as well as gram negative bacteria. Now students, Why we are using this word positive and negative? There is some reason for which we are using this word positive and negative. This positive and negative word was given by the scientist named Christian Graham. Okay. He has isolated this bacteria on the basis of the technique Gram staining. Okay. The name of the technique was Gram staining. After his name, he has put the name Christian Graham. Okay. And after that, he has concluded the name Gram positive and Gram negative bacteria. Okay. So in next class, I will teach you Gram staining procedure, how the technique has been done, and how we are distinguishing it on the basis of Gram staining procedure as Gram positive and Gram negative bacteria. Clear with it, students? So with this, I am
Yes,
ending
ma'am.
today's lecture, students.